Welcome to this research story about photoreceptor microsaccades. Most insects have two main visual organs, called the compound eyes. They are composed of lens-capped units called ommatidia. Underneath the lenses, photoreceptors' light-sensitive parts, called the rhabdomeres, convert light into an electrical signal that goes deeper in the brain. But breakthrough research has showed that photoreceptors do more. When exposed to light flashes, photoreceptors themselves contract and deform without any muscle. In the eye, these contractions cause rapid rhabdomere displacements called photoreceptor microsaccades. They enable the flies to see much finer details than previously believed and perceive depth. Despite the recent advances, much about the photoreceptor microsaccades remain in dark. We wanted to investigate how photoreceptor microsaccades spread in the compound eye, a very important question. Based on our previous findings, we expect that the photoreceptor microsaccades are localized, meaning that they won't spread up far away from the active receptors. To test this hypothesis, we set to image photoreceptor microsaccades from 3 locations, while only activating the receptors on the blue area here. The yellow receptors not seeing the flash should stay still, and the forward-facing receptors that see the flash should move. Next, we designed the experimental setup. We based it around our previous deep pseudopupil imaging system you see here. The fly is on a pipette tip. And we have 2 rotation stages. Things get interesting under infrared back illumination. We can use computer simulations to see what is going on. These are the rhabdomere tips. Under infrared illumination they appear bright, but we cannot see them because of the lenses. Real microscopes have a narrow depth of field. Let's focus deeper inside the eye. Bit too much. This is the deep pseudopupil. It is a magnified virtual image of the rhabdomere tips that we saw earlier. And when the rhabdomeres move, the deep pseudopupil follows, allowing us to measure the rhabdomeres' movement. But back to building the new system. To secure parts in their places, we used an aluminium profile called maker beam. Its T-slot design allows fast prototyping and rigid structure. We used it to build these frames. Then we took 2 3-axis manipulators and mounted the new cameras on those. And we were ready. We hypothesized that a flash delivered through the microscope would evoke photoreceptor microsaccades on the receptors that see the flash, but not on the side-facing receptors that look elsewhere. Strangely, however, the flash evoked photoreceptor microsaccades visible in all 3 cameras, despite the side receptors not seeing the flash. This result was unexpected, but luckily there was a reasonable explanation for it. The flash illuminates the fly and the pipette tip, making them to scatter light. Some light reaches the side cameras and is reflected back to the fly. We can simulate this. Can you spot the reflections? Here's one. From the fly's perspective, the flash also comes from the side cameras, explaining our results. To overcome the reflection issue, we added a light source over each camera. From this angle, mainly the reflections would activate the image photoreceptors. We hypothesized that by using different stimuli for each light source, matching movement patterns would emerge in the corresponding cameras. And despite the lens reflection crosstalk between the sides, we could correctly identify the used stimulus from the deep pseudobubble movements. This result indicates that the photoreceptor microsaccades in the left and right eye work independently. Photoreceptor microsaccades are fast movements caused by sensory cell contractions. They enable flies to see their surroundings in high detail and perceive depth. This research enhances our understanding about the compound eye vision and has practical applications in the field of robotics and imaging technology. Thank you for watching.